Hi, I'm Valentin Lestringant, cytogeneticist at the Hematology Laboratory of CHU Amiens Picardie, and I will present to you our work on the use of bionano genome optical mapping in a series of ALL cases. To begin, we choose to focus on AL because of the frequency and structural diversity of cytogenetic anomalies in these pathologies. The characterization of these abnormalities remains a central point in the management of patients with ALL by their prognostic and therapeutic impact. This diversity requires the use of several conventional and molecular cytogenetic techniques to establish the most exhaustive genetic profile. And so we investigated if optical mapping had the capacity to replace all standard techniques. Our objectives were to evaluate both analytical performance of the optical mapping technology and computer tools for data processing and filtering. In summary, are operators able to easily isolate anomalies of interest from other signals? We studied 10 AL previously genetically typed that were selected to obtain a representative panel of major prognostic impact anomalies and their structural diversity. Optical mapping data were interpreted independently by blind operators before comparison. And secondly, we investigated the possibility of detecting copy-neutral loss of heterozygosity. Selected cases were 10 B or T ALL treated in Amiens. Reference techniques were performed in Amiens and Rouen for karyotype and FISH, and in Lille for SNP array and RT-MLPA. Optical genome mapping was performed using bone marrow DMSO pellets and the platform Gentian in Clermont-Ferrand. The rare variant pipeline was used for the analysis. And blind operators were Joyce Lee, bionano engineer, and myself. About the selection of anomalies, in SNP, we retained all anomalies with a size superior of 1 MB and or involving a list of 186 genes of interest involved in leukemogenesis, and with a minimum size of 20 kb. For other techniques, all the anomalies found were selected. And for optical mapping, the anomalies were first extracted according to the size and confidence score criteria, and then selected using similar criteria as in SNP. About patient characteristics, we had 6 B ALL and 40 ALL, 7 children and 3 adults. Using standard techniques, we found a total of 80 anomalies meeting inclusion criteria, and we excluded 2 CN-LOH that were separately analyzed. Using optical mapping, after extraction, we obtained a total of 948 calls. After filtering and analyzing these calls, 89 anomalies for the first operator and 84 for the second were retained. In total, 73 of the 80 referenced anomalies were found by optical mapping. 7 anomalies were missed, and 12 anomalies not described by standard techniques were newly found by optical mapping. Here are the anomalies correctly found in optical mapping. As you can see, the majority of anomalies of clinical interest were found, including, as examples, hyperdiploidy, 21 intrachromosomal amplification, and some recurrent translocations. Here are the anomalies that were missed by optical mapping for reasons that I will detail. In the first case, trisomy 3 and trisomy 4 were missed, but clearly visible in the raw data using the whole genome CNV representation. The 3Q region was more gain, which allowed it to exit filters for data extraction. In the same way, 22 gain pretty visible in raw data was missed in another case. Subclonal Y chromosome gain 22 was missed too. It was found in 7 of 22 metaphases in karyotype, but quantified at 7% of nuclei by fish. So this is below the detection limit of optical mapping and suggests an in vitro proliferative advantage of this subclone. Here is the case of a near tetraploid subclone that was found in 6 of 19 metaphases, where close to tetraploidy with a variable number of chromosomes, but with a low resolution at the limit of the classifiable. It was neither found by SNP array, so that may suggest a true tetraploid subclone. Two anomalies were missed because involving a pseudo-autosomal region. This includes a deletion leading to P2LY8 CRLF2 fusion. Pseudo-autosomal regions are, as other regions with highly repeated patterns, like centromeres, telomeres, are indeed not easily accessible using optical mapping, as well as other techniques. To finish this VPRB1 deletion was correctly found by the technology, but missed by operators during the analysis, as it was hidden by recurring deletions of the IGL locus. Here we are showing anomalies newly found in optical mapping. 3 of them consider rearrangements well described in ALL, leading to LMO2 TRA fusion, TRB-MIC fusion, and IGH CEBPB fusion. This translocation involved various breakpoints and led to partner overexpression, so no transcript could be found using MLPA. However, fish are non-systematically performed on these targets, so we decide to confirm them using fish probes. Now, as an example, the case of the LMO2 TRI rearrangement. This case was actually more complex because involving rearrangement of TLX1 as well. Here you can see the different derivatives, the blue maps from rearrangements of TLX1 with TRA at the top and LMO2 with TRA at the bottom. These derivatives align with the reference maps in green, with chromosome 14 at the TRA locus at the bottom, and at the top with chromosome 10 and TLX1, or chromosome 11 and LMO2. Note that crossed lines in LMO2 derivatives symbolize an inversion. As shown by the pink dotted lines, 2 breakpoints are present in the TRA locus. The first one in the middle of the gene was shared by the 2 derivatives of the middle, and the second on the right was shared by the 2 other derivatives. So if we schematize chromosomes, we have a total of 4 breakpoints, one in the chromosome 10 involving TLX1, 2 in the TRA locus on chromosome 14, and another one in the chromosome 11 involving LMO2. Interchromosomal translocations then resulted in 2 derivates from a pretty classical 1014 TLX1 TRA translocation that was found by karyotype. And the TRA fragment between the 2 breakpoints will be inverted and inserted into LMO2 onto chromosome 11, resulting in a LMO2 TRA rearrangement. Here we show the fish analysis using the LMO2 probe that shows no split signal, and the TRAD probe showing a typical split signal corresponding to the TLX1 translocation and an extra green signal compatible with the insertion into LMO2. Regarding other newly found anomalies, we have 2 other translocations to control, 2 high complexity rearrangements, and there remain 6 deletions, 2 gains, and 1 duplication. 4 of them involved no gene of interest but were higher than 1 MB and only found by the CNV tool. Turned out to be false positive signals. The remaining anomalies were confirmed by the SNP array raw data. CN-LOH detection, which is not possible by optical mapping in theory, was investigated using the de novo assembly pipeline. We removed all filters, including database variants, to show all variants detected by optical mapping. Then we looked into how much variants were called homozygous by the SV tool in the LOH regions previously detected by SNP. As you can see, in the 2 samples who carried the CN-LOH, the rate of homozygous variants was above 90%, which was pretty much higher than the rates in these regions for patients free of CN-LOH or deletion. To finish, it's important to mention that we faced a problem of cross-sample contamination. 17 anomalies were in fact exactly the same between sample 4 and sample 7, with very low molecule counts for the contaminated sample, which is sample 7, and including a BCR-Belson translocation. And this makes us consider the implementation of a system to identify DNA molecules, such as a code borrowing system. To conclude, we found the bionano optical mapping technology to be pretty mature. It provides a resolution and analytical sensibility as expected and promised. Remaining defaults are anomalies occurring in the pseudo-autosomal region, centromeres and telomeres, as well as gains and losses of chromosomes in small fraction. Cross-sample contamination also remains a problem to be solved. General use in diagnosis obviously needs larger cohort trials and needs to be tested in other hemopathies, but overall optical mapping is already a promising tool for research of new variants and for the resolution of high complexity rearrangements. Thanks to all the people who contributed to this work, and thank you for your attention.